So I'd like to talk today about some of my very recent work, a new area for me, which is using some mathematical and computer modeling techniques to look at the morphology of the Ediacaran fronds. So, okay. So by fronds, I'm referring to these large, so centimeter to even meter scale organisms with this frondose branching morphology. So these are the sort of core data set here of mine. On the left have been grouped as the rangeomorph. It's like rangia here. They share this very fine scale branching. There are also some other frondose species, such as these two within genus Charniodiscus, which also have a similar frondose shape, but the very fine scale branching is not preserved in these specimens. So some of the aspects of their morphology, such as this frond leaf-like shape, stem, basal holdfast that you see in some of these species, and as well as this fine branching itself, are reminiscent of other taxa. However, there's nothing quite like them known today or at any other point in the fossil record. So they're, in this sense, lost constructions, as Silaca put it. And there are a lot of questions about their morphology, their affinities, their life habits, and their evolution. So one possible route into understanding them a little bit better, I thought, would be a more detailed description of their morphology. So here's a slide illustrating this fine-scale repetitive branching, which has been described as fractal and approximately self-similar, because it's repeated over 3 orders. So we see here these primary branches here are divided further into secondary branches and then smaller tertiary branches from these. Although other authors have noted that this self-similarity is, of course, finite, and it's also, it seems that some species look more self-similar than others. So we might ask, what exactly is self-similarity, and can we quantify it? So in a self-similar branching system, we can have the same rules for branching, which are applied repeatedly. And as a result, subparts map to sections of the whole. So this primary branch here maps to this top section of the whole branch. But we can also describe partially self-similar systems in which the parts don't map perfectly to other sections. But they are overall similar, but there have been a number of edits or changes that deviate from self-similarity. So the questions that particularly interested me were, can we use a self-similar branching process to model these Ediacaran fronds? And then if this is possible, can these different species be modeled within a unified system? So can we get one system with multiple parameters that can have different values and in combination describe these different morphologies? If we can do this, we might be able to then ask how self-similar, or in contrast, how complex each morphology is. And this might tell us something about their evolution and the adaptive function of these otherwise lost constructions. So the model that I've developed incorporates a number of general features that have kind of been agreed upon and can be observed across multiple species. So it's an alternate branching system. So this means that, so for example, in this specimen of Charnia here, you can see that up the long axis, we have these large primary branches that alternate in this sort of left, right pattern. And so in the model, we see left branching, right branching. And so these branches develop, the same process is applied iteratively within these sub-branches. Okay. The model also includes apical branching. So this means that the free ends of the branching system will be the sites for growth, for further branching in the next step of the process. So this is supported by observations that in most species, the smaller branches that appear to be new growth are located on the apices of the branches. We can also include internode expansion. So we can have segments that grow as well as branch. So this means that these older internodes here within the branching system can expand so that they take up a greater proportion of the length of the main axis. So this allows us to start to model some of the changes in spacing of branches that we can observe in the fossil specimens. And we can gradually start to try and increase the realism of the model by adding more parameters. So we can include, for example, branching delays, so that the branching of lateral branches here is delayed relative to the main axis. These can change the shape of the overall branching system and the sub-branches themselves. We can use cylinders. This is supported by observation of those very beautifully preserved fossils that have sort of micrometer scale preservation. So we can have cylinders that increase in length and also diameter as they age. And then we can take this branching into 3 dimensions. Okay. So importantly, we can also have targeted growth parameters. So this means that we can vary growth or branching for segments that have a different age or a different branching order. So for example, we can change the growth rate in the main stem relative to the primary branches. Or we can have the primary branches leaving the stem at a slightly different angle from the secondary branches on the primary axis. Okay, the complete model which I've developed has 30 parameters that describe these things, branching and growth rates, and allow a targeted parameter setting for the stem, the primary, secondary, and tertiary branches. Of these, 24 parameters were required to model the species that I have in the data set that we saw in the first slide. Okay, so with this model, I think it's possible to create quite realistic representations and reconstructions that capture quite a lot of the morphology that we can see in the fossil specimens. For example, this beautiful Beothucus specimen here. So if we go into this model in a little bit more detail, we can I can explain how this was developed. So in this representation, the segments are colored according to age. So we can see that the oldest segments are here at the base of the stem, and that the youngest ones are these apices, so colored in blue. So this is the youngest part of the main axis, but all of these blue sections are the same age. Some major features of this species and its model are a rotation of the primary branches. So I interpret this as a 90-degree rotation so that we only see one side of the branch from in frontal view. There's also, we can incorporate curvature, so there's negative curvature here of the primary branches, and then positive curvature of the secondary branches, as well as a slight helical torsion to the stem. Now this is one of my favorites. So this Fractal, this interesting spindle-shaped form. So this is the more elongate species. It's Fractal misrae. So this illustrates how the model was informed by preservation of the fossil and incorporated those preservational features. So, for example, we can see here in this very nice specimen that the fossil shows that the organism was folded over. So we see the long axis here. This section here seems to have been folded over this other section. And this shows something interesting. So you can see there's a slight 3-dimensional curvature, but it appears that this was curved on both sides. So you have positive curvature. It's interesting how we can, how can we explain this identical morphology? So my interpretation of this is that there are a number of rotations to an alternate branching system, so that we have the branches then overlap. So we're looking now at front view onto the specimen we saw on the previous slide. And this gives you this morphology here. There's a longitudinal axis in the center, but it's not in contact with the sediment here. This species is interpreted to be a basal sort of benthic recliner lying on this side here. So the stem is not usually preserved. As these are impressions, this is compatible with this morphology. Okay, one of my conclusions is that we can use this model, which is a unified model with the same set of parameters, to describe what are really some, by appearance, quite diverse morphologies. So for example, this is my model of Charnia versus Fronds, another sort of elongate, possibly benthic reclining form. So if we have a look at those in a little more detail, we can see in Charnia, I've interpreted the morphology to be formed by another 90-degree rotation of the primary branches so that they're upturned. They also have a curvature which curves upwards. And as a result, each primary branch is nested inside the one below it. And this produces this characteristic zigzag morphology up the long axis, which has been considered for a long time a diagnostic feature of this genus. So Pectinofronds, this morphology is achieved by taking a duplication and rotation of the system for branching. So this is, we have younger segments here, but also here. And these older ones are of the same age, with the addition of a number of rotations which bring up the primary branches to lie in this sort of comb shape. So this is compatible with Brazier and colleagues' suggestion of there being two poles of growth, which was suggested for Fractifusus. I interpret this to be a feature of both Fractifusus and Pectinofronds. We can see that these species do have differing levels of self-similarity. So, for example, if we look at these two, we can see Avalofractus here has a highly self-similar morphology in which the primary branches are an almost perfect copy of the whole frond. Actually, no edits are required to the branching parameters for the separate orders of branching. So I consider this, in terms of its branching, to be perfectly self-similar. Whereas in other forms, there are modifications of the parameters for the different orders of branching. I suggest that this is to do with the diversification in space-filling strategies in the group. So for example, in Trapacia here, we have, as I interpret it, primary branches which are held very close to the stem, but then a difference in the branching order for the later branches, which allows them to both exploit height first and then expand laterally and in depth. So having got this model, I'm now looking to what we can really pull out of this. So I'm working on cladistic analysis of the parameters to look at the affinities between these different frondose organisms and explore the evolutionary transitions in their branching architecture and self-similarity within the group. I'd also like to look at the disparity from the different assemblages. I'm exploring some alternative branching models, as for some of the species, there are some alternative decisions that could be made about the branching parameters, and I'd like to see how that affects the results. And I'd also like to explore the relationships between those groups, which have been described as rangeomorphs, and some other frondose species. Okay. I'd like to thank my colleagues at University of Cambridge and thank people who kindly let me look at specimens. Of course, our funding body, image sources, and thank you for listening.